Well, good morning. Sorry for the delay, but we're glad that you could join us this morning and listen to some of our research projects that are sponsored by the Mississippi soybean growers through the soybean checkoff funds. We're excited that our Mississippi Soybean Promotion Board could sponsor these projects today. And we have many more that we would like to share with you at a later date. So just look for those emails as we move forward through this program. I'm Dennis Reginelli with the Mississippi Soybean Promotion Board. I'm the Executive Director. And just want to cover a couple slides before I turn the program over to our first speaker, Dr. Mary Love Taggart and Mr. Blake Hodges. Just want to inform all of you that we have an outstanding web page, mssoy.org. We have some very good information there that you can research. We're proud of our variety selector tool. We have research reports from all scientists. We post the annual and the final reports on this site. We have a couple outstanding guides, the scouting guide, the irrigation guide that you can also download from this site. We have social media opportunities, Twitter, Facebook, YouTube. So check out the mssoy.org and follow us on our social media pages. Again, we have 5 speakers today, and some of these projects have been completed, some are ongoing, and we will offer certified crop advisory credits. So if you have the little scan app on your phone, at the end of the program, you could just put your phone up to the screen and you should get credit for participating in this program. If you do not have the scanned app, I would suggest that you go get it from the App Store for Certified Crop Advisory and go ahead on and download it. If not, just email me, Dennis R. at Bell South, with your name and certification number, and I will make sure that we record your name as completing the course. This program is being recorded and will be shared at a later date. And we will hopefully give you credit. If you participate at a later date, we will give you credit for the certification. We thank you all for joining, and we look forward to the great presentations today. And at this time, I'm going to turn over the program to Dr. Mary Love Taggart, and she will start us with the first slide.
Good morning. Can everybody hear okay? All right, we're going to start off by talking a little bit about infill soil moisture variability and how that affects our sensor placement in the field and also irrigation. And I just want to recognize my co-presenter, Blake Hodges, who's the graduate student who worked on this project. He just successfully defended his thesis recently. And then I also want to recognize the co-PIs on this project, Dr. Joel Paz and Dr. Ming in geosciences. I want to, of course, thank the Mississippi Soybean Promotion Board for funding this project, and also the Southern SARE Graduate Student Fellowship for funding some of Blade's work. This first slide shows the 10 mid-south and southeastern states and the number of irrigated acres in each of those states. And we can see this data came from the Irrigation and Water Management Survey, which used to be called the Farm and Ranch Irrigation Survey. But since 2002, we've had an increase in the number of irrigated acres in all of these 10 states except for in Texas. And if we go to 2017, we see that Mississippi has the 3rd largest number of irrigated acres of all these 10 states, behind Arkansas and Texas. And the reason for that is that we have less rainfall during the growing season combined with periodic drought. So this map on the right-hand side of the screen shows Northeast Mississippi, the counties in Northeast Mississippi, We can see that even though we receive an annual average rainfall of about 56 inches, we only get about 30% of that during the summer growing season when we need that rainfall to grow a crop. So these counties you see in East Mississippi, that region also contains the Blackland Prairie region, which has historically been in dryland production just due to the high cost of drilling wells. We have to go down sometimes to 500 to 600 feet to reach water. And then even then you might drill and not be guaranteed to reach water. So, but more producers in this region of the state are irrigating using surface water and center pivots. And the rolling topography in this area kind of lends itself to that. And you can see in this map, the blue dots indicate locations for surface water permits, and the maroon dots indicate locations for groundwater permits. So you can see that we've got, and this is a few years old, so we've got an increasing amount of surface water use in this area. So when we're using surface water, a surface water irrigation pond, we have a finite amount of water that we've got to depend on to get us through the growing season to use for irrigation. So it's really important to use water efficiently and not use any more than we actually need. And we know that soil moisture sensors are a relatively inexpensive tool that can help determine when to schedule irrigation applications and conserve water. These sensors are granular matrix sensors, which measure soil tension or soil water potential from 0 to 239 centibars. And they measure that through resistance of an electrical current between two electrodes. 0 centibars would be when the soil is completely saturated, and 239 centibars would be extremely dry. So the overall goal for this project was to try to improve our recommendations for sensor placement within a field. We see there are different publications out there that have different recommendations. Watermark instructions recommend a minimum of one set of sensors for every 10 to 15 acre fields. Some areas recommend 1 to 2 sensor sets every 80 to 100 acre fields. We also wanted to look to see if we could identify less invasive or surrogate methods for estimating soil moisture to improve irrigation efficiency. So the first objective was to measure infill, spatial, and temporal variability of soil moisture in the active rooting zone of the crop using watermark sensors, and also look at the correlation of root zone soil moisture to other variables like soil texture, leaf area index, and plant height. We also used the DSAT crop model, but we don't have time to discuss those results today. So we started this project in 2018 in the Blackland Prairie region in Noxubee County near Brooksville, Mississippi. We're working on an 18-hectare field that's irrigated with a center pivot, and they're in a soybean and corn rotation. So they grew soybeans in 2018, corn in 2019, and soybeans again in 2020. We used ArcGIS to create a 55 square meter grid, which resulted in 44 grid points under the pivot. And you can see those black dots. The blue circle is the outer edge of the pivot, and then the maroon, between the blue and the maroon, that indicates the area covered by the end gun of the pivot. Soybeans were planted on May 3rd of 2018 and May 4th of this year, and then corn was planted on March the 30th of 2019. And we had a watchdog weather station in the northeast corner of the field. We used a Trimble UMA to navigate to our points with a 2 to 4 meter accuracy, and we also had a Lindsay GrowSmart magnetic flow meter to monitor our irrigation events. So at each of those 44 points, we installed 2 sensors, a set of sensors, one at 12 inches or 31 centimeters and one at 61 centimeters or 24 inches. So one set of sensors, 12 and 24 inches in 2018 and 2020 for soybeans. And then in 2019, when we had corn, we put in an additional sensor at 30 inches. We cycle those sensors according to the watermark instructions and install them wet with good soil to sensor contact. And then we connected those to a watermark data logger that you can see there in the blue in the right image. We also recorded leaf area index with a LAI 2200 plant canopy analyzer, and we measured plant height an average of 3 measurements weekly for each growing season. So that resulted in 9 sampling dates in 2018, 12 dates in 2019, and 10 dates in 2020. Also, the first year of the project in 2018, they harvested on September the 19th, and then we went in the following day and pulled soil samples to measure soil texture. We pulled 2 to 3 cores for each one of those 55-meter grid cells, and then combined those into a composite sample. And we pulled those cores at a depth of 24 inches because we wanted to be sure and get the soil texture in the active rooting zone. Now Blade Hodges is going to talk about the results from Objective 1.
Good morning. So in 2018, in these first 3 graphs that I'm going to go through, we'll see kind of the same trend for all 3 seasons. In 2018, we can see that these lines represent the root zone average for each set of soil moisture sensors across the field. And as they increase on the y-axis, their soil matrix potential is increasing, which means the field is actually drying out. And as we go throughout the season, we see a lot of ups and downs with irrigation and then rainfall. In 2018, we didn't get that much rainfall in the beginning of the season, so we actually see where we started averaging the sensors together on June the 15th, which was pretty early in the season, because we saw roots had reached the 24-inch depth. In 2019, we see that there was a lot of early season rainfall, so we actually didn't start including the deeper depth sensors until later in the season, later in the growing season. And we also see that even though there's a lot of rainfall in this season alone, during that time period, we got 27 inches of rainfall, which is, like Dr. Taggart mentioned, that's half of the average rainfall during the growing season. So even with that huge amount of rainfall, there were still periods of drought where we needed to irrigate, and we probably wouldn't have known that without the sensors. And then again in 2020, there was a good bit of rain early in the season, and so we didn't average that deeper sensor until July the 20th. So we see that the root depth plays a big role in how we know when to irrigate and when to trigger those irrigations with that root zone average. Here we just see that the sensors that fell outside of the pivot that were only covered by the end gun were not receiving as good a coverage of irrigation. So we actually just took those out of our analysis in the rest of the presentation. So here we have the density of, we have different schemes of density of sensors, and we use inverse distance weighted interpolation methods to predict the soil moisture across the field based on the data from each point. On the first graph, we see the elevation management zone, and it's based off of the elevation. We put those 4 sensors, those 4 points in there based off the elevation, and then the rest of them are just gridded schemes to see what the results were from a gridded scheme of soil sensors throughout a field and at different densities. So we started with large grids of only 4 sensor sets and then worked our way all the way to the original 55-meter grid that we had with the 39 sensor sets in the field. So on June the 26th of 2018, this is what that would look like if we ran those interpolations for each different scheme, but just that one day. So we see that we would have made the same recommendation, which would have been to irrigate, and which they did do, based off of the orange and red colors all over the map, which the above 60 centibars for center pivot irrigation was our trigger. And then, but we also see that on the elevation management zone, we do get a representation of a low spot in the field where it is wet, as opposed to the gridded scheme that only had 4 sensors where it would have shown that the whole field was dry. And we'll see how elevation played a part in this in a little bit. But also our goal the recommendation of to irrigate or not to irrigate is based off of a uniform irrigation management. So the uniform irrigation application. So we also have to take into effect, is the producer going to be applying a uniform application, or is he or she going to be trying to use variable rate irrigation? And in that case, we would start off with a higher density of sensors, like the 39 sensor sets or fewer, but at any rate, a higher density, and then scale that back down after we find the pattern in the field that the soil moisture has spatially. So these tables show the inverse distance weighted results, the agreement between the model and what we actually measured at each of those points for each scheme on 6 different days throughout the year. And we found 3 dry days and 3 wet days. Usually it was before and after an irrigation event. And so we would compare the dry versus a wet day and highlight which grid cell had the higher R-square value, or the highest agreement with the model and what we measured. And in 2018, most of those days actually fell on the wet days, but in 2019 and 2020, most of them fell on the dry days, which is good because that's when we would like for our model agreement to be the highest, to be able to be more critical in our timing of irrigation events. So in conclusion, for objective 1, the variability across the field is highest when the field is driest. The more dense the array of sensors are, the more accurate the soil moisture predictions will be using interpolation methods. And for 2 out of the 3 years, the IDW model predictions were better on dry days the majority of the time. And then the data collected in this study present a case for creating site-specific management zones for the implementation of precision irrigation. The next objective was to evaluate the correlation of root zone soil moisture to soil texture, LAI, and plant height. So as I mentioned earlier, we saw where the soil moisture was quite variable across the field at times, and usually we would think that that had something to do with soil texture. So we checked that out, and the field was quite homogeneous with about 5 of these grids classifying as silt loam, but even in the soil texture triangle, you can see that they're still very close to the silty clay loam ones. And so the soil was quite homogeneous across the field. And then we tried to see if LAI or plant height played a big role in being able to predict soil moisture. And plant height had the highest correlation to soil tension all 3 years, even though it wasn't a very strong correlation, with the R-squared being below 0.5 all 3 years. And then we looked at the elevation and how it played a role in the soil moisture. And here's a digital elevation model. You can see it goes from high to low as you go from the east to the western side of the field, with a little bit of rolling topography in between. And here is a picture from each 3 seasons, and we can see that the same moisture pattern, the spatial pattern of the soil moisture, took place all 3 years as the drying happened throughout the season, leading up to an irrigation or a rainfall event. We can see that it is more variable as it dries out, and that the pattern was similar all 3 seasons. And then we see how the yield was affected by the soil moisture. On the 2018 yield, we can see that low spot in the eastern or the western side of the field having an impact on that yield being lower. And then we also see a valley in the southwestern quadrant where the yield was lower in 2019. And actually, when that early season rainfall came, it washed out a lot of that and had to be replanted. So that definitely affected the yield in that season. So in conclusion of objective 2, neither of the 2 vegetative characteristics measured had a strong correlation to soil moisture, but plant height did have the strongest out of the 2. Soil moisture variability can occur despite a homogeneous soil type across the field, and elevation plays a big role in variability across the field, especially in this part of the state. And the drying pattern was similar in all 3 seasons, which could be critical to future findings. And now I'll turn it back over to Dr. Taggart. So
I just wanted to wrap up with some take-home messages. one of the things that was kind of surprising to us, we expected a little more heterogeneity in the soil texture. However, that was kind of not an issue in this field because we did have a very homogeneous soil type. It did not contribute to the variability in soil moisture in the field or in the modeling results. However, we did see, as Blade mentioned, that elevation seemed to play the biggest role in soil moisture variability here. So it's really important, even if you're looking at a small field, when you're considering sensor placement and density, you've got to look at all factors that might be contributing to that variability on a farm-to-farm and field-by-field basis. And this study does reinforce the need to utilize all that knowledge of the field's characteristics to make an informed decision on sensor placement, especially with soil texture and elevation. So we know that soil texture does affect soil moisture variability. We just didn't have that issue here in this field. So even for a small field such as this one, 44-acre field, one sensor set is not going to likely be enough, especially when you've got a change in elevation or soil texture. So this study provides guidance on how a grower might determine placement and density of soil moisture sensors for uniform irrigation or variable rate irrigation. And I think this study also presents a good case for variable rate irrigation. So the more heterogeneity you have in a field, the more likely that producer is going to benefit from a variable rate application. And that concludes our talk. I don't know if we have time for questions or if we need to pass it to the next presenter.
We should probably move to the next presenter and perhaps take questions at the end.
So I'll stop sharing now. And I think
Dr.
Jesse, are you
Wilkerson.
next?
There we go. You are on mute if you
Oh. Sorry. Okay, so I'm Tessie Wilkerson. I'm a pathologist at the Delta Research and Extension Center. Today I'm going to talk to you a little bit about one of the big concerns, which is grain quality or soybean grain quality. On certain years when weather is conducive, and even in years that we wouldn't think there would be, there's issues with quality at harvest when plots are left in the field or your crop is left in the field. And so growers struggle with this issue. So we're trying to come up with some type of solution or reasons why this occurs. So I wanted to go ahead and thank Mississippi Soybean Promotion Board. They have funded this project for several years now, and we're getting there, but we're not there yet. So we really do appreciate them continuing to fund them. Also do receive some funding as well from USB for some work on seed quality. So I also want to recognize them, and also have co-PIs, Maria Tomaso Peterson and Suja. I'm just going to call, I'm not sure how to pronounce her last name, but she works at the water center here as well. Let's see. Well, for some reason, my slides are they're not advancing. Let's see. Okay. All right. Let's skip those. I don't know why I added those. Okay. So anyway, so in seed quality, when we're looking at the symptoms, it will include shriveled, elongated, moldy, chalky seed. And we usually have reduced germination, which causes an issue when you're producing seed for production, and also issues with vigor. Currently, our management strategies that we have in place would be crop rotation with non-hosts for fungi that we think might be causing issues, fungicide application, although we don't, even though there is suggestions that this might help, we don't really have enough data to support or negate whether fungicide applications help with this issue. And then also one of the biggest thing is resistant cultivars. And information regarding which of the cultivars that are commercially available have resistance is often limited. So we're actually working towards trying to get that information out there as well with some state variety trials that we're looking at with delayed harvest timings. Mostly, and historically, we've always thought that this is environmentally dependent during warm and humid conditions in advanced growth stages, basically when crops left in the field and the farmer's not able to get to it for a certain amount of time, we do see increasing damage. So we're trying to find a solution to this, and what is actually causing it. Okay. So what I'm just going to show you is some pictures of some seed quality, some seed that you might see in the pod. These are actually some pictures that Tom Allen took. You can see phomopsis, when you see these like whitish, chalky looking seed, that's because there is phomopsis infection, purple seed stain, and also some other things that are causing rotten seeds in there that we're not necessarily sure what exactly it is, and that's kind of what we're trying to find out. Okay, some economics behind this. Some years, such as 2018, we did see that phomopsis did cause economic losses in both southern and northern states. And according to Southern Soybean Disease Workers, 52,000 metric tons were lost across 60 of the southern states. So 0.2% of all economic losses as a result of this. So on a big scale is a pretty good chunk. So it's definitely a concern. And if you're looking at dockage, which is anything over the 2% is going to get you some reduction at the elevator. So that's a pretty important thing for producers. So for this research, we have several objectives. One of the things that we're doing is collaborating with the USDA breeders to test some of their breeding lines that they have bred to have minimal seed damage. Also to determine the fungal organisms that come off the harvested grain, which might be assisting in reducing quality. We're also going to look at some fungicide sensitivity to the organisms that the fungi that we get off the seed. We're looking at, as a result of stink bug feeding, are there more fungi on the seed? We know that with increased feeding that, we see more damage, but we want to know why. Is it something to do with a particular fungi that might be on the seed coming in? We also want to determine the fungicide efficacy. So when we do spray, is this really helping reduce seed damage? And so with that, we were looking at spraying some stuff late growth stage. And then also if we spray on time and also with delayed harvests. Okay, so I'm gonna talk a little bit about our methods. So this is our field at Stoneville. We have 2 shelters that are covered. They're both equipped with overhead irrigation. We have, so in 2018, 2019, and 2020, we had trials under both shelters. And in 2019 and 2020, we incorporated 2 trials that are identical to the ones under the shelters from the request of some board members wanting to know, so if we have these lines under here, these breeding lines, what do they do if you just put them in a natural environment? How do they respond? So we have 2 trials outside identical to the ones under the shelter. And we also incorporated an insect trial to look at stink bug feeding. And so under these shelters and outside the shelters, we have 21 lines. 18 of those are PI lines from USDA breeders, and 3 of them are commercial checks. We have these planted in May of 2018 July 2019 and June of 2020. Unfortunately, we were not able to get them all planted in the very same time, but due to weather and some issues with our shelters, just how it turned out. And so also when we harvested, we would hand harvest these 2 weeks prior to R8 of each line. So there are some lines out there that are group 3s and some that are group 4s. So we would harvest 2 weeks past the R8 point to simulate that delayed harvest. So we have these plots set up as a randomized complete block design with 3 reps. We inoculated all these plots with Phomopsis, which is historically thought to be the cause of the seed damage at R5. And we begin overhead irrigation at R5 to conclude with about 200 hours underneath the shelters. And then of course, natural rainfall in the outside trials. And then we applied fungicide to one of the shelters and then one of the outside trials at R7. So I'm going to go through some of these lines with you. So the 3 in bold are, of course, the commercial checks. The cadens and the progeny are both susceptible, which they're calling susceptible to damage. The 6181, which you'll see in when I get to the data, is the resistant check. It's Phomopsis resistant developed to be, and then also the Pella 86, which is going to be your susceptible check, and all the other lines in between are some of the offspring, so to speak, of the resistant lines. Okay, so moving into the data, this is going to be, I'm not going to go through all these numbers, but this is going to be your plot weight and your visual damage on a 0 to 10 scale. We also have a lot more data, including Mid-South grain data from these plots, and also germination, kernel weights, things like that, but it was way too much to put on a slide. So today I'm just going to talk about plot weights and visual damage. And you obviously can see that on one side you have the plot weight, and then the other is damage. And what you can see is from plot weight, we're obviously the lines are not there yet as far as plot weight when compared to the commercial checks. But if you look over at the damage, you do see that some of the lines are promising when compared to the commercial checks, including 093, which is located in down the line there with some pretty low damage scores. Okay, and this is looking at the same type of data, but this is under the plots that have, under the shelters that have fungicide applied at R7. And you don't see much benefit when you're adding in the fungicide treatments as far as plot weight or the damage, but we do continue to see a trend in damage reduction with some of the breeding lines, such as the 093 again. And even though we still see damage in the Phomopsis resistant lines, it leads us to believe that there may be some other organisms involved or other fungi involved. And that's what we are also working on this project, to see what's in there, what else is causing it, since there is still damage in the Phomopsis resistant lines. Okay, and so this data is going to be from the trials. We only had these, of course, in like 2019 and 2020 on the outside. This first slide is going to be the non-fungicide treated plots located in Stoneville. Set up the same way with your 3 susceptible commercial lines, and then your lines, your breeding lines. We obviously didn't see the conditions in 2020 if you look at the damage ratings that we had in 2019, but we see that we do still see more differences between the lines when the conditions are more conducive for damage regardless of. So like in 2018, we had 22 inches of rain, 2019, 20, and 2020, 17. But regardless of the lines, we still do see damage in these plots, even in years when we don't have as much conducive environment. right, so this next data is going to be from the outside plots that were fungicide treated. Again, we didn't see much benefit from the fungicide, but interestingly enough, in some cases, there was more damage in the fungicide plots, which leads us to believe that there's something could be being knocked out with fungicide treatment, something that's beneficial out there that's controlling something else that might be contributing to damage as far as maybe some beneficial fungi or something else. So we're still trying to figure some of these things out. Right. So moving into the insect data, I don't have 2020 in here yet. This is all 2019 data. But as we can see, obviously there's more injury from when there's insect feeding. But I do want to say that what we're going to do is we're going to take this seed and we're going to take these to the lab and do some assays to see what fungi come off these seed as compared to the other plots that were managed for insects. And these were obviously infested. As you can see, when the cages, we left these on there, allow the stink bugs to feed, to put pressure and see if there's any differences in the things that come off the seed when we move to the lab. We also think that if you look at this there might be something going on with these lines that some of them maybe were attractive to the stink bugs. We're not sure about that, but that's something that might be looked into further later. And something that we added in 2020 and we'll continue to add in is this little small trial that we did with some with one fungicide, Moravis top, where we included applications at R3, R6, and in combination, and then a desiccant at the end with these combination applications. And as we set these up as 4-row plots in order to harvest each row at different timings. So every 2 weeks prior to the R8 to see if there was differences in damage with these particular plots as well. And we'll also send this to Biz South Grain. So this data will be pending and ongoing as well. We actually still have some plots left in the field. So, okay, and so moving on. So from this seed that we get out of the field, we'll move these into the lab. Through some molecular techniques, we'll determine which fungi are located or which fungi are present on this seed. And then we'll take from that, we'll also do some fungicide sensitivity assays in which we take the fungi that we get and subject them to different fungicides to see which ones are sensitive and if there are some that are less sensitive at different amounts. And so this is just some preliminary data that we have identified. Several things that are several isolates, or several fungi that we've taken off the seed. This will be ongoing. This is only from 2018, so we still have 2019 and 2020 to get through. It takes a lot of time in the lab to get these done, so this research will be ongoing. And so just kind of like how this fell out. Interestingly enough, of course, we see Phomopsis was present in both the non-fungicide shelter and the fungicide shelter in 2018. But interesting enough, we see that Cornespora was also pretty high. We found a lot of Cornesporas as well. So it'll be interesting to see as we go through more of this, what all we find. And maybe do some combinations of some things later to see how they react together. So in summary, some level of harvested grain damage occurred regardless of variety. We've identified 6 fungi on the seed. Cornespora was one of the most prevalent. And we do see some level of damage with commercial varieties, damage tolerance with commercial varieties. Like I said, we are working through some of the OBT stuff that Mississippi State puts out to let everyone know which ones we have found that were more resistant than others. And we definitely need more research on this. We're not there yet. So stay tuned, there's more to come. And so we're getting closer. my colleagues at the USDA have said that they would like to release some lines with the lower DKT by 2021. So that's promising. We do have a better understanding of what crop protection practices can be used to manage and reduce seed quality. And we're working on updating existing management recommendations and extension publications to be able to get this information out there. Some outreach efforts that we're going through. We do have information available on the Mississippi Crop Situation blog. Also, Crop Protection Network has some information out there on our research, and we are training graduate students and trying to get, the future researchers looking at some of these things so that we can find a solution to this problem. And I'd like to acknowledge Dr. Tom Allen. He also works on seed quality. And of course, my co-PIs, Maria and Suja. We have graduate students, and of course, couldn't do any of this work without our support staff and our summer workers. They are wonderful and help so much. And then want to thank, of course, my funding sources. And I can take any questions or we can move on. So.
Great, thank you. I think for the sake of time, we might go ahead and move on. So if you want to stop sharing your screen,
okay.
And then perhaps we'll take questions at the end as long as everyone is okay with time. Andrew, whenever you're ready, you can begin. You're on mute.
Sorry, it's not showing up in the right screen yet. Let me see if I can get that. Is it still, what does it show up for y'all? Is it good?
We now see it full screen.
Okay, good. All right. Well, I'm going to go. There we go. I'm sorry about that. So just kind of jumping in for my talk and the supported project from the Mississippi Soybean Promotion Board that I'll be discussing today is really two different focuses that we'll be looking at, but it is the automation and evaluation of automation and new technologies that are available in specifically in furrow irrigation. Going out, just see what's out there that we can put on farm. So all this is really on farm focused, and then the second focus of this project, funded through the program, which is our Riser program, which really focuses on outreach, extension, and on farm evaluation. And going into that second focus is. So we have the first is the investigation of the new technology, and then we have some proven technology and proven practices and honing in on adoption of those specific practices that we know that have worked, and then the research has proven that they can save time, water, and money. And so with those, I'm going to go through and really wanted to cover both aspects of that. And so hopefully it's not too much as we go through it on that, but I think both of those aspects are important. So what we're trying to talk about, whether we're talking about new technology and evaluation and or promoting a proven technology, we want to make sure when we talk about sustainability that we have that weighted balance between all these sustainable agricultural components, and that's economically viable. We want to make sure that it makes sense from the bottom line for the producer. If it's not going to save money or make more money, then we're all wasting our time on that end of it. And then we want to be able to, can we do things to save time, same labor that'll help the farm families and the quality of life. And then the sustainability from the environmental sustainability side of things. Can we save that water? And we're going to try through this program, we're really trying to focus on all of those and make sure we hit all those aspects of that sustainability. And so this is just the first year of our on-farm evaluation and trials through our evaluation of the automated system. And so a lot of this is just a lot of trial and error as we figure out. I think we've got our feet below, settled down for the next year as we go forward, and how these things need to operate, and how we need to go through the system. And that's what we wanted to do. We wanted to evaluate these new technologies and how can they be work into an operation system, a farming operation. Do they work? That is the main objective. When they open and close, are they going to do what they say and they're supposed to do? And then as we step forward through that process, then we can do some more evaluation on total water savings, and then that economic profitability. Does it pay for the investment into these things? Will it save that time and labor to make it worth from the economic sustainability mindset? So what is, when I say automation in furrow irrigation, and really what I'm talking about is being able to control the irrigation pumping station. And that is simply a pump control where we can turn it on and off. And then at the same time, we have valves. And so depending on the number of fields or number of sets irrigated from that well, we can control valves. So we have actuated valves that can open and close. So we can transition through fields, close off, and move on to the next set or the next field through controlled valves. And then lastly, we are able to utilize that off soil moisture sensors. And it was a great being able to follow up what Dr. Taggart and has done with the soil moisture sensors and how we can use those for scheduling. And so with that as our trigger to get the process started, we can start the program. And so again, growers' ability to control pump station and then complete what I'm calling that spin from the very beginning to the end of that irrigation remotely. And then can it save time, water, and then money? And so what components does this consist of? There's actuated valves. And so each one of these valves have an actuator on that can open and close. And we have a single valve, and we have and you can see there we have a T or a double actuated valves, or each one of those can be open or closed at the same time, or we can close them completely off if we need to go off to the next field. And then how they're all communicating together within this system. We have our soil moisture sensors that have a one-way reading to our telemetry system. And so it's just going to give us readings to our telemetry box into our system. And we have our actuated valves that I just showed pictures of. And that's a two-way communication. It's going to tell us when they're open and closed, but at the same time, we can control those. We can open and close those through the system. And then we have our irrigation pump controller. Same thing, it can communicate. We're going to tell what the status is, or we can open and close that. And it's going to tell us what the status is, whether it is open or closed. So that's a two-way communication going into the internet, stored on the server. But really what this does is it gives it back to a user interface to where we can, I guess, relatively simply control what's going on from the pumps to the valves based on irrigation or soil moisture sensors, and based on those triggers that we want to set in. So here's just a quick image of what it looks like as far as what our schedule may be. And so these are solar valves. You can name them however you want, whether it's a field, whether it's a set, and then you set your run times and you set your orders. And through that, so we have our run times and we have our orders. And then once it goes through that spin, it'll operate from the very beginning of the field to the last one. The ideally is seamlessly, so that as it transitions over, there's no over or extra watering until the person gets back to switch it over, or whether that comes, at 11 o'clock at night, and the thought is, well, I'll get it first thing in the morning. And so you reduce those extra number of hours that the water could be running. And it's based on soil moisture sensors that we know where we want to set our triggers. So here's just a scenario of what that could look like. We have our riser, or we have our well and the riser at each one. And so as we water out, we set our set times. As it goes through that, the set one's going to water out. And then as it reaches our number of hours of irrigation, the valve on field 2 will open. And as it opens, it'll begin to water, and then field 1 then closes off. And then the same thing as you go through your spin, field 3 will then open. And then once it starts, it's open and it begins to irrigate, field 2 will then close, and so on as we get down to through field 4. And then once it ends, it will, obviously complete our irrigation cycle. So just, kind of going through a scenario, depending on set times and depending on when you start, if you ideally want to start this one at 7 a.m. on this day, based on that set time, it's going to go to 11 a.m. the next day. This one's going to be a 24-hour set. So you're going to be 11 a.m. finish time on the next day. And then we get into, off times where, okay, now this one ended at 3 a.m. What are you going to do? You're not going to come out there at that time. So we can transition at that next field over to field 4 at that time and finish out. And so, I mean, you can, depending on the number of hours and set as you go through, those are some scenarios where you can, whether we're triggering on saving water, but just because we're triggering on soil moisture sensors, because that's going to save us water in a sense. But also, if you're doing everything else right and using all these practices, can automation even close that gap even further on the total water use? And again, this is year 1. It is preliminary evaluation, and really we're focused on does the valves open and close when they say we do? When we set a schedule, does it operate correctly? We had some issues, but as far as controlling the valves and them opening and closing, we had no issues. I think we had more, if anybody spent any time on a farm operation, many things are going to go wrong that are outside of this system, but that are affected by this. And so, I mean, it could be a wire that got ripped up, or we had, this is all dependent on electricity if we're doing a pump control through electric well. And so that's another thing to consider when rural electricity goes in and out during a spin cycle, that's going to throw you. And so those are all things that we saw that first year. One component that I didn't mention, and it's a very important component on there, if you can see, if you're able to see my cursor or not, but there's a blowout valve right there. And so that's going to be our weakest point. And so if for some reason we have all our valves closed, then that's going to release the water through there with a lot less pressure resistance than say underground pipe or any of the valves, so that if something goes wrong, we have a release point with that. And the other thing we tried on the left side, because if you do blow out that valve, then water is just going to be pouring out of there until it gets fixed or until you cover that blowout back. And so we had this stacked pipe here to where if something happens, it releases based on our pressure, our height, our PSI, it's going to go out the top. And then once things get opened and situated, it can go back down into that stacked pipe, and you can continue on without having to go and replace that blowout valve within that. And we started using that because one of our situations that we had a water pressure within the underground system within a certain site. We didn't have it anywhere else. That just would really have a lot of push through the underground system and would blow off all of our, because when we control, instead of shutting off a field at a hydrant, we're shutting off on the outside of the hydrant at the valves. And so there was too much pressure at the hydrant that would blow off the top of our valves on one particular site. And so with that, we had this, that's where we got into the stacked pipe. We released that initial knock or that initial pressure. Once it blew out, and then it all settled back in, that water level dropped, and then it continued to operate. So that was one adjustment workaround that we did based on one of those systems. And so again, year 1, there's within 3 sites on our soybean fields, this is what we saw with the reduction in water use. 10 to 6.7 in our acre inches, relative to the same yield. So, but again, as we go forward and we're continuing to evaluate and to continue to put more sites and more numbers with that, then hopefully we'll be able to evaluate those numbers and again, not only see what the production or does it work? Does it function correctly? But then, and then going into from a standpoint of recommendations, adoptions, do they work? And then the economic standpoint, which is going to be a really important with things like this, that does it pay to operate this, or maybe there's certain situations where these can be utilized the best, especially from depending on a grower or irrigation decision maker, how many sites that they have, how many sites are they controlling within that operation, or is there one site that's far out there that takes a lot of time to go through and back and forth and changing out those valves. But we've got to have confidence in, once we start that spin, does it open and close all the way through? And so a lot of good progress as we're going through with that. And then transitioning really completely over to more of an extension and outreach focus. What we're doing is, in the past, we've done these on-farm evaluations, and we know some of these practices that do work, and they've been proven. And so we can go to the step of, this is what works and this is what we recommend to adopt. And so this is really focusing the second portion of this project objective is to focus on adoption of soil moisture sensors. Using soil moisture sensors, surge irrigation, and computerized hole selection has shown to save to save water, to save time, and to save money. And so what you can see, profitability is increased. We're saving a lot of water. And so for this, we feel real comfortable that these are some adoption standpoints that we want to focus on. And one of those three, of the surge sensors and computerized hole selection, is what we wanted to focus on specifically with this on-farm demonstration. And I say it's beyond on-farm demonstrating. It's really working with the grower, not just setting it up and operating it, but having them a part through the whole process with that. And so we focused and honed in on the soil moisture sensors. And so from the same survey that Dr. Tackett showed, is what are the methods that farmers are looking at for scheduling irrigation, and by far it's going to be condition of crop and visual condition of the crop, and then fill of soil. Then we go down to soil moisture sensors. So some of the common issues or maybe hurdles or barriers when looking at soil moisture sensors is interpreting the sensor data. A lot of tons of information, a lot of numbers that get spit out from that. And what do we do with those? What do we do with these numbers? Just putting a soil moisture sensor in our field is not going to save water. We have to utilize that information. And so not only taking that information and understanding what it's telling us, but then what information can we do to, and that goes into our triggers and our starting off our irrigation sets. And then how do the different types of data, different types of acquisitions of those data. So the manual telemetry services, systems cost, those are some of the things that we see as barriers as we go forward with that. So we're trying to address some of those that we think that can be easily addressed with that. And so what our, again, I called it on-farm demo, but we're really working one-on-one within these individual farmers that have not adopted soil moisture sensors yet. So we're going out on their farm, and then from that process, from installation to the reading the data, then we're interpreting the data with them, sending them the readings, and then using that data, helping them set up triggers for their specific field. And then at the end of the season, we'll pull those sensors, and we have an end of the year meeting to where we go over the graphs, what it looked like, and how they operated through that system. And so things that we're working with them on is, one of the things that we have that I've noticed that we have a lot of maybe it's a terminology concern or issue with that, is triggers versus thresholds. And so we have science, we have research that shows where our thresholds are. And we don't want the soil moisture sensor or the soil moisture to go past a certain threshold. And that's where we see yield reducing with that soil moisture within that field. But that doesn't mean that's when our trigger is. And so we need to back off that threshold so that we're done irrigating before our field hits that threshold. And that trigger is going to be dependent on how long it takes you to get around, well capacity, size of fields, and all those things. And so that was a big information gap that I saw as we went through this process, is when to trigger based on the numbers and that we have. And so that's what we're working with. And this trigger depends on their current situation. And so how we can go through that. And then we talk about that trigger, that threshold, It's going to be different throughout the growing season. So our water use based on growth stage within the soybean plant is going to be varied between those weeks. And so if you're scheduling on a calendar schedule or on a weekly basis, the same way as you do R3 and R4, but then you go into R5 through R7, it's going to be a difference there on total water use. And so you may be over irrigating, and especially in this area, this back end where it continues to drop, we can really push our irrigation frequencies quite a bit through that when we utilize soil moisture sensors, because it's using a lot less water. So we're working with them irrigation guys within that system. And then things that, trying not to go into detail, as we work through and some of the barriers and issues that we see is soil moisture sensors in the field, and then how we read those data, but then how we read individual sensors, because as the growth stages go through, we don't need to be reading the deeper soil moisture sensors because the roots aren't there yet. And so how do we, when we get that information, how we read it, and then what do we need to focus on reading? So we're going to go through the growth, the season with them, looking at these soil moisture sensors, where here in this case, we're using the 6 and the 12 inches, because that's where our roots are being utilizing that depth of that water. And then as we go through, we can start employing the third sensor. We start to see it extract water. So let's start putting it into our equation as we look at those readings and as we weight that out. And then we can take our readings and our Cinnabar readings from that equation, and then going forward, and here's our fourth sensor. And so I think some of these things is working through the farmer to make sure taking that information, and then what sensors to read based on the year, or specifically the growth stage of that plant. And so we're setting that up, we're walking through them, and then we're showing those recommendations. And then really whether they use those or not within this project, and especially on the first year, has varied. And so we're really working with them on how whatever their pace may be within that, because this is going to be a multi-year project that we stay on their farm, because we're not seeing the jumping right into it on some of them on that first year. And so this kind of goes back to the triggers versus thresholds that we wanted to talk to with them. And then with that, we're going to work with them on, look at the set hours, the total spend time, and then working back from our thresholds that we want to set. And then we can come up with when we want to start that irrigation with that trigger. Here's some examples of our sites this year that we saw, and this was one that listened well to our thresholds. And with those thresholds that we set for them, in this case, we went just a little bit above 80 because that's where we felt comfortable with them. We think we could push it a little bit more, but we wanted to go with them and set that little comfortable zone for that. And then that bottom line is our trigger. That's when we wanted them to trigger the initiation so that we wouldn't pass that top bar of that threshold. And so you can see this is our weighted average up here. And so we really never, even this first one, we never crossed our threshold. And then we settled in, and then when we triggered on triggered the irrigation on our trigger, and then we irrigated here, and as we go up, sorry to explain the graph, but the higher it is, the drier the soil moisture is. And so with an irrigation or rainfall, we're going to see that saturation and wet as it drops down through our Cinnabar readings. And this was a very good season with this grower, very comfortable with it. And this particular one, they're ready to go and buy in. And so we work through the process of what soil moisture sensors they want to use. And then on the other side of that, we also had one that we went through the same process with them, but you can see, so here's our threshold up here, and then really triggering most of the irrigations down here around 20 to 30, which is really wet as far as the field goes, and then just irrigating a lot more frequently. You can see the number of irrigations compared to that, and we could really spread these out, especially as we get into this R6 time frame, as it's using a lot less water. It's not drying out as quickly as it would be earlier in the season, but continue that irrigation scheduling continues to be the same with that. And so we, with this particular grower as well, we sat down, went through the same graph with them, and working through the different ranges and meeting each grower where they're at and comfortability with themselves, but with the technology, and then pacing that through that system. So I think we've got a good plan for next year on this particular grower, to where some of the decisions, we're going to push them up hopefully a little bit, and next year we can have a prettier graph to show that we're pushing up a little bit and a lot less irrigations. Kind of wrapping that's the on-farm demo, but just I wanted to be able to show the links here, that some of the things that we're working on, just as resources, not really going into it, but just point out resources that we have. And so through our National Center for Alluvial Aquifer Research, or the Water Center, we have a website that helps with this, that I use, that the Soybean Promotion Board has helped support this soil moisture sensor showcase, where we have all the available technologies or telemetry services for sensors that are out there, because that's a common question we get, is what type of system do I use? What type of telemetry service do we use, or what's out there? And so instead of having to go through and purchase them on their own and not like it, or even waste a whole entire season and not like it, we gave them the most common services that are out there, to where they can go on this site and then click in a username and password, click on that, and work through and see how the information is presented and what feels most comfortable for them, and how they can make those decisions. They make different types of graphs, they weigh out the readings, and then you can set your thresholds and triggers within each type, very differently within each one. So what fits their personality or their system or their operation. And then we go through each one of these, whether it's a one-on-one consultation with them, or it's just out there that they can click on and work through, to just alleviate the uncertainty of which one to use, and kind of leaping out there and making a guess. And here's a site of that's more of the online interaction, which is really where it's focused on. But we also have the hardware. So different ones are going to offer different hardware out in the field and different obstructions as they operate through their system. So that's another way to visually go out and take a look and see what that looks like within the variability within each of these soil moisture sensors. And then within that, also at the same site, you can click on, we created a lot of videos to where we can, how we operate each service, each telemetry service, and then some installation and removal type of videos to help, again, close that gap of information that they may be wondering about, those barriers that we see of uncertainty. And so we can have those videos to help answer a lot of those questions. And lastly, we're focused in on a lot of extension fact sheets that we can work through and give to the grower, or go through the county extension office, that specifically answer a lot of these questions, again, that we see with soil moisture sensors, whether it's how do I take the measurements, what are my options there, can I make my own sensors, where do I put the sensors, and then how do we install it, and then how do we schedule irrigation off those. And so with those, as we work through those, I think the top soil sensor irrigation triggers, and then I believe locations is where we're at now. So these aren't all up yet as we work through this and get them posted, but they'll be up on the extension website, and again, that Water Center website as well. With that, I just want to thank the Soybean Promotion Board for the support of this project, and then through the on-farm demos, especially with that's really done through the county extension agents, and so utilizing them to be able to find farmers that are interested but haven't adopted yet, and really being the interpreter for those data that they're being sent, and then every other day or weekly readings and suggestions are being sent to the county extension agents to these farmers. So I appreciate the effort with that. And that I can take, I guess we'll wait for questions at the end. I'll stop sharing. Thank you.
Excellent, thank you.
All right, so I guess I'll go ahead and get started. Obviously, I'm not Dr. Jason Bond. My name's actually Hunter Bowman. I've been working for Dr. Bond for about 2 years at Stoneville towards a PhD. And a little bit about myself, I grew up on a cotton and soybean farm near Marianna, Arkansas, just across the river. It's about 30 miles north of Helena, if any of you know where that's at. So today I'm just going to give you a brief update on a few projects that we've been working on for the Soybean Promotion Board in Stoneville to just address some of the recent weed control issues in soybean production for the past 2 years. So this slide here just sets up the way the past few years have gone for us, the way we've got started. Whether you're dealing with water being on the field or crop residue left in the field from the previous year, maybe the fall was too wet to mow your stalks or deal with your ice stubble. The field may be simply too wet on one half to spray the entire field. You can only spray half the field when you're running your burn down, or it's just taking so long to get in there that you're spraying weeds that are simply too big to get good coverage or control of. The next slide here just reiterates what I was saying there, where you've got weeds that are too big. You're dealing with winter annuals later in the year than you normally would, summer annuals earlier in the year than we normally would. April 4th is pretty early for us to be thinking about what we're going to do about palmer emerging. So now with the wet years we've had and the wide adoption of the Extend technology in Mississippi, the majority of questions that we've been answering are, what are my options for prickly sida, or many people know it as tea weed? Why can we not kill barnyard grass, and not just only barnyard grass, but other grasses as well? And even in some cases, what's the best option for terminating a stand of, say, corn or even soybeans before we come back and replant? So the first thing we're going to talk about here are prickly sida options. And the data here just shows levels from herbicides applied prior to the emergence of that prickly sida, so at the time that we plant our crop. And the point here is that, we have a lot of options for prickly sida control pre-emergence. The thing is, when you're thinking about this prickly sida, you can't forget about your driver weeds that you're dealing with, such as like Palmer amaranth or some type of grass species. So when we look at these same herbicides for control of Palmer amaranth or barnyard grass, what you see is that we're pretty good at controlling both of these weeds. However, we do need to utilize 2 effective herbicides to do this. And as you can see, anything that contained dual or even command for grass control, and then say something like metribuzin for control of that Palmer amaranth, gave us pretty good control of all 3 weeds if you think about these 2 graphs and the previous graph that I showed. So just show you what that looked like in the field here, you got to remember this is a month out from these herbicide applications. As you can see with these single herbicides here, we control different weeds. However, we don't control all these weeds simultaneously. So when we look at those herbicides together, what you see is actually, better control of all 3 weeds. Now, the thing you've got to remember when you're looking at these bottom 2 pictures here is this is a month out. So you're probably getting ready to come back with like a post application of something else. So that's what we're going to get into next, is our post options for control of that prickly sida. And this graph here is it's set up by technology, where you've got your Roundup first, and then Roundup Ready, Extend, Liberty Link, and then your 2,4-D. So the first thing that you probably noticed by this graph is that when we add Python to one of those first 3 technologies, we can pick up control of prickly sida, and we can increase that pretty well. The second thing that I noticed looking at this data is that 2,4-D is more active on that prickly sida than dicamba would be. So when we look at these same herbicides for Palmer amaranth control, you're probably not very surprised by this. The main take home here is that if we use Select Star, dicamba, Liberty, 2,4-D, it doesn't really matter what you're going out there with. You're probably going to have to make a follow-up application to get acceptable control. Now, I'm not going to show you grass control for these herbicides like I did with pre-emergence herbicides, but we're going to get in that pretty more in depth here in just a minute. So here, just show you what that was like. So as you can see, when you add that Python to the tank, you pick up control pretty well. Not much different though from that Liberty or Enlist 1 or 2,4-D. So now to get into the grass control section of this, this is actually one of the main things that I'm working on in my PhD project. So lately, a lot of the questions we've got with this STEM technology is, why are we not killing barnyard grass? And the way we've approached this is we're looking at it in a number of different ways of when we mix dicamba and Roundup, what's happening? Is it application equipment, or is it GPA? Is it timing of application? So just to look at that here, first graph I'm going to show you is actually just looking at nozzle type. So this is going to be pulled over all those different herbicide treatments, whether it's Roundup alone or Select or Roundup plus dicamba or Roundup plus Select or all three of them together. What you see is that when we go to that TTI nozzle that's required to make those dicamba applications, you actually lose about 5 or 10% control. This is because it produces a more coarse, larger droplet and reduces the coverage that we're getting. Now, when you look at the different herbicide treatments, the main thing you'll notice is that the more things we add in that tank, the less control that we're getting there with those herbicides. So just here to show you what that looks like. It's hard to see with these pictures, the level of green, the difference from all these herbicides. But when you're standing in the field, it's very noticeable that the more you add to that tank, the more green you're going to see after that application. Now these pictures here just show the difference from the tips we use, from your flat fan to an air mix to a TTI. Now that's much easier to tell the story here with these pictures, that as we go to that more coarse droplet, we're getting reduced control of that grass. So the next thing we're going to talk about is a question we got some is, does formulation of Roundup affect that level of antagonism or the reduction of barnyard grass control we're seeing? Can we use a different formulation or maybe a different distributor and achieve better control? And the best answer I have to that question after the past 2 years is no, that formulation is not really playing a role in that. The only reduction that you're seeing is when you do and add that dicamba to the tank. One thing about this, I don't know if this is going to matter much going forward, because with the new registration of dicamba that was just approved, they actually removed all IPA salts from being able to be tank mixed with it because of the increased volatility. So here, showing you that you've got your glyphosate alone and your glyphosate plus Enlist and glyphosate plus Ingenia, and the main thing you notice is that just adding that dicamba or even 2,4-D to the tank just reduces the efficacy of that glyphosate. So the next thing we're going to talk about is a question we actually got last year in one of the county meetings is, and we hadn't actually thought about this until this question came up, is when we add residuals to the tank, is that also hurting the level of control that we're seeing? And, last year was the first year that we looked at this, and looking at it now, my answer is that we do not have a definitive answer to this. You do see some reduced control here at 14 days when you add dual or say warrant to the tank. Now, I don't have a graph showing you 21 days, the level of control that we had, but I can say that that mixture where warrant was included, it increased. And the reason I think that is because warrant actually uses an encapsulated formulation of acetochlor. So it's released slower, so it took longer for that to take effect. So here, just showing you that out in the field, what you see is that, number one, we grew some pretty good soybeans, and number two, the only difference really was that Ingenia in the tank. There's not much difference from just adding a residual. So the last project that I'm going to talk about that we've been working on is actually a project that a master's student started on this year, Alex Mangialardi. And what he's looking at is what's the best option for controlling a failed stand of soybeans or even corn, and how long after do you make that application to control that do you need to wait before you get back in the field with the planter? So the first thing you're going to look at here is a graph of how long did we wait to make these applications after the soybeans merged. So you've got 3 days after the application of the different timings after emergence and 14 days. And what you see is that the problem is that we have to give these soybeans time to get big enough to control them. If we come in there, we decide, hey, they're not coming up, the seed's rotting in the ground or something, we're going to go ahead and spray and come back and replant. Well, that may not be your best idea, because what actually did come up, you got to give that time to get big enough so that you can get control of it and get good coverage. So here, that's probably hard to think, well, I've got to wait 10 days to spray these soybeans that I'm going to kill anyway. And then I got to wait another 10 days for the herbicide to take effect. So just to show you a picture of how that can help you out, here, even at 5 days, you can see the increased control. We also, get better activity with gramoxone if we add, say, tricor or, metribuzin, even just waiting 5 days after emergence. Pretty much prior to 5 days, as you can see, you're just wasting your money just going out there. So just to show you a little bit more about that with that gramoxone, or when we add that metribuzin to it. So this is just 3 days after we made these applications. What you can see is, at 10 days is when you really start to get good control of those soybeans. So if they've been emerged 10 days, you add gramoxone, metribuzin in your tank, you're probably okay if you just wait 3 days. So you're looking at 13 days from the time that soybean emerged and you made the decision you wanted to replant before you actually get back in the field with a planter. Now, if you want to go with something like Roundup and Select, you're looking at a little bit longer. You don't see the difference in time after emergence, so you might be able to get out there a little earlier, but you still got to wait 14 days. So you're looking at about the same timeline there on before you re-enter the field with that planter. And here, just going into the corn, what you see with that gramoxone or versus the Roundup and Select. Either way, you're waiting a lot longer than anybody wants to wait before you can replant soybeans. You're already upset, you lost your corn, you're having to come back and replant. Nobody wants to wait 14 to 20 days before they can get back in the field. But if you don't, what you're going to have is basically corn that becomes a weed out there in your soybean field later in the year. Just a few more pictures of that to try to bring that home. The longer you wait, the better it's going to be, the cleaner it's going to be, the more activity you're going to get out of that herbicide application. So if you didn't pay attention to anything I said, just go ahead and take a picture of this and it'll pretty much give you everything you need to know. If we're only controlling one weed, we're going to allow infestations of other weeds. That's what happened with the Extend technology. We were thinking about Palmer amaranth, and now prickly cider or tea weeds blown up on us. Prickly cider, it requires additional herbicide modes of action beyond those required just for the Palmer amaranth, as we've seen. And summer annual grass control can be influenced by our spray nozzles or what we're putting in the tank. And what we're even seeing now is maybe some application volumes. If you've got guys that are trying to put out a lower GPA so they can cover more acres with the tank, that they might be losing a little bit there. So if you think about that, if we lose 5 or 10% because we have to go to a TTI nozzle, then we add our dicamba to the tank, we'll lose another 5 to 10%. Well, then say you cut your GPA 2 or 3 gallons per acre just to cover a few more acres, you lose another 5 to 10%. You've given up 25 to 30% of your potential weed control before you ever entered the field with the sprayer, not to think about the environmental conditions that could come up and also reduce that control. So just being mindful of that before we even get to the field can really help with that grass control. Another thing we added this year that I didn't show was we came back and did follow-up applications with Roundup and dicamba at different timings after that initial application. And pretty much the summary of that is, whether we spread Roundup alone or we put dicamba in there in that second application, we pretty much got above 95% control of that grass. So it's going to take a second application of Roundup to get grass in these Extend technologies. Looking at the replant situations, timing is important. Choice of herbicide treatment, is going to be important with managing those failed stands. With soybeans, you can go with paraquat and metribuzin. My thing about that, looking at that with paraquat and metribuzin versus Roundup and Select, I don't know personally, but I was just wondering, if you're doing that and then you're waiting 14 days or so before you plant, you're going to have to get back in closer with those post options. So that's something to be mindful of. And then also, what's that planter going to do in the drill passes where, to the herbicide layer? Are you going to have weeds coming up in that drill pass? So that's something to think about if you're putting your metribuzin out prior to getting in there with the planter. While everybody's here, just going to throw this out there. With the weed management book this year, we actually added a peanut section. So there's a rating table there now for your options in peanuts, different weeds, the efficacy of those. And then also within the other crops, we broke the post control out by technology. So you can go to that crop, go to the post, and then figure out, if you got Roundup, Liberty Link, Extend, or Enlist, what your options are. It'll give you ratings for that, rates for different soil types and everything, just to help everybody out with that. I guess we're not going to do any questions right now. I'll try to answer any we have at the end. If not, there's Dr. Bond's phone number, and I'm sure he could answer them. Thank y'all. All right, I'll
Thank
stop.
you very much, Hunter. Okay,
All right. You hear me okay?
we can.
Good deal. Well, I know we got started a little bit later with some technical issues, but I'll try to get through this pretty quickly and get everybody along on their way. So my name is Trent Irby. I'm the Extension Soybean Specialist here at Mississippi State. And I've got several, I guess, different projects that I'll talk about today, but all of them are conducted under the SMART program, which is a very large demonstration effort that's funded by the Mississippi Soybean Promotion Board. So let me first say a special thank you to the Promotion Board for sponsoring this project, as well as the other projects that you've seen today and those that you may see in the future. Mississippi has done a really good job in the last several years, really, in terms of our soybean production systems, and it's a major commodity for us. And I think a lot of that's due to the efforts of a lot of these projects. So on that line of conversation, I guess, let me just give a quick update for 2020. 2020 is 2020. But on a positive note, let's just look at what our crop has done this year. So we were up 430,000 acres estimated from last year. A lot of that is due to the acres that were flooded out in 2019 and went unplanted. The big thing, I think, though, was our yield. We got the crop in the ground this year. We didn't have the earliest of planting dates, but things weren't super late either. Didn't have a whole lot of issues earlier in the year. Late in the year, we battled some pest issues, depending on where you're standing. Weather kind of got hot and dry in some places. And of course, we dodged or went through quite a few hurricanes, depending on where you're standing. But overall, they're currently predicting our state average yield at 54 bushels per acre, and that would be equal to our previous yield record. I think that's a big deal, and I've made the statement a few times, and I stand by this. I think that if we hadn't seen that hot, dry period that we had sort of towards the end, that took its toll on a lot of our dryland acres. So if we hadn't seen that, I believe we would have shattered our previous yield record this year, just in terms of the way things looked. So Here's an outline of what I intend to cover today. I'll start with some fungicide application and yield. So for the purposes of this presentation, I'm not going to focus on the disease ratings or disease data. These are some projects that really, this is 3 different projects. So this will be some graduate student projects that were conducted in 2 different places, 2 different students. The first one was done in 2017, 2018. The next data set would be 2019 and 2020. And then of course, I've got our on-farm fungicide demo program results that I'll show from 2020, which is something that Dr. Tom Allen, our extension plant pathologist, and I started several years ago, looking at large plot fungicide application with comparing different products to untreated soybean to just measure, on a big scale, what kind of yield response we will see. So first, this graph that you're looking at now, this is fungicide application timing. This was a graduate student project that we looked at various products applied from R3 to R6, and even had a split application where we had the first one done at R3 followed by a separate application at R5. We didn't see a significant response to the application timing, but we did see a response to the product. So again, for the purposes of this presentation, we'll just focus on the yield. And I guess what I'll just point out is it kind of goes with what we've been seeing, I guess, over the last 4 or 5 years. We have a couple of different multiple mode of action products that we used in this particular project, and that would be a tank mix of Prexor and Domark, or an application of Quadris Top SBX in this case, and compared that to a single mode of action of Quadris or an untreated check. So, I don't know, 5, 6 years ago now, we began to see a lack of response from applications with just single modes of action like a Quadris. And I think a lot of that's due to the resistance issues that we've seen. But in a nutshell, we did see a significant yield response to the 2 tank mix or pre-mix with multiple modes of actions, and those range from right at 3.5% yield increase. Next, I'll look at this is a project. This is actually a project focusing more on seed quality, but this is the fungicide application component of it and our yield response to those. So these were Mirvis Top, Prexor, and Domark tank mix. Again, Quadris by itself and an untreated check with an application timing at R4. And this was done in 2019 and 2020 at several locations around the state. And again, we see, a 4.5 to roughly 6.5% yield increase from these automatic fungicide applications. Next, I'll show the data. This is from our large plot trials from 2020. The maroon bars that you're looking at are our irrigated locations that we had, and then the gray bar was a single non-irrigated location that we had this year. We weren't able to separate these yields out statistically. We see just a 1 to 2, 2.5 kind of bushel response when it's averaged over all the locations. But in a nutshell, this is what we've been seeing for several years now. When you look at our yield responses to these fungicide applications, certain locations we see big responses, and some locations we don't see any response. So there's obviously, this first bullet point there, we don't always see a statistical yield response. But when we look at these over the yield values, I guess, it averages between 2.5 to 3.5 bushels per acre is what we see for these positive response scenarios. So when you put the economics behind this and look at, does it pay for itself? We do see a positive economic return for these automatic fungicide application. However, it does depend on the total application cost, which would be, obviously including application method, ground versus air, or the product that is selected. And then probably more importantly, that depends on the market price that we have for soybeans. So I know we've seen some fluctuation in that. That conversation is a lot easier to be had if we go back up to a higher market price. Next, I'll spend just a minute talking about harvest aid uses in soybeans. Traditionally, I guess we use harvest aids mainly to take out late emerging weeds. to facilitate a more efficient harvest with the machine. Over the last 7 or 8 years, we've really made this application more so to speed up the drying of immature plants and to be able to get the machine in there and get the crop out a few days faster compared to waiting on it to naturally senesce. And then obviously we have issues with green stem on an annual basis. And when I say green stem, there's really 2 different kind of ways to look at that. One is a genetic green stem where that plant just will not shut down. That's most often a case of the genetics when it's on a big scale. We do see some serious green problems from insects like red-banded stink bugs. And then we'll also see just some natural variability out there. And I think a lot of that's tied to kind of the emergence and where you stand just on the day of maturity of that crop. So different scenarios, I guess, for green stem. But we have a handful of products with labeled uses as harvest days and beans. The main 3 that we use here are paraquat, sharpen, and sodium chlorate. There's the rates and rate ranges, the application timings according to the labels, and the pre-harvest intervals for each of those products. So we've done a good bit of harvest aid work over the last several years, just looking at performance of different products, looking at application timings of the different products, and even looking at some adjuvant systems within the different products that we have. So today I'm just gonna show a performance evaluation, looking at an application of paraquat by itself. So that would be our standard rate of paraquat by itself, or a tank mix of paraquat and sharpen, or a tank mix of paraquat and sodium chlorate, which is also a common treatment that we'll see, and then a standalone application of sodium chlorate and a standalone application of sharpen. One thing that I'll point out is the application rate of sodium chlorate when it's tank mixed with paraquat. We'll recommend that it goes out at 3 pounds per acre. If you find yourself in a situation using just sodium chlorate by itself, it would need to go out at a higher rate, at 6 pounds of chlorate per acre. Sharpen, the rate range is 1 to 2 ounces. In the work we've done, we found that 1.5 ounces is as good as the performance from a 2 ounce rate. So that's why we use that in this particular evaluation. So I won't spend a lot of time on this graph, but just showing the percent desiccation that we would see. Whenever we do harvest aid performance evaluations, we'll look at it 3 days after application, again 7 days after application, and then we'll do a final rating at 15 days after application, just to kind of see how each product performs over time. So in this particular case, I'm just showing the 3 and the 7 day. The 3 is in the black bars, and the 7 day after application is in the green bars. You'll notice that products like paraquat, which tend to act a little bit faster than sodium chlorate by itself or sharpen, 3 days after application, we have a pretty high level of desiccation, but I'll show you some pictures in a minute showing the general greenness that's still out there in the stems and in the plant. Sodium chloride I mentioned is a little bit slower, and then sharpen 3 days after application was even a little bit slower than that. But what you'll see is that by 7 days after application, it didn't matter what product we had, they had all dried down and reached a level where it would feed through the machine. So again, just a few pictures to show. This was what the field looked like at the time of application. And then as I cycle through these, I'll show you on the left-hand side would be the picture that was taken 3 days after application, and then again, 7 days after application. And like Hunter mentioned, it's kind of difficult to see from these pictures, but if you're walking through that field, you would notice that the leaves have all crisped up and fallen in a lot of cases, but the stems and the petioles, there's still a lot of green in that 3 day after application picture. Here's paraquat with a sharpened tank mix, paraquat sodium chlorate. This is chlorate by itself. You notice a little bit more green in the 3 day after application picture. And then here's 1.5 ounces of sharpened by itself. And again, much more green 3 days, but by 7, it's ready to put a machine into. And one of the reasons to show this, I guess, is sometimes I'll get calls on, we made this application and it's still a lot of green. It won't feed through there. Through the machine real good. The leaves have all crisped up, but the stems and petioles are still a lot of green. part of that's just time, and environment plays a factor in this too, temperature, humidity, and things of that nature. If we jump out and try to get into it before it's ready, it may appear ready from the turn row, but once you get out in it and start cutting it, you'll see some green in those stems. And I think that in a lot of cases, when it's big, broad field scale kind of scenario, that is just simply time. More time is needed for that crop to go on and change on and dry on down. But the bottom line is in all the work that we've done, paraquat, sodium chlorate, Sharpen, all of these products are good, equal products after the appropriate amount of time went by, which in this case, we found that by a week after, everything was good to go. But I will point out that Sharpen and sodium chlorate were the only ones that met their pre-harvest interval this quickly. So another big question is, how early is too early to spray my harvest aid? There's been work done, in different places, different states. Previous research out of Louisiana suggests that paraquat applied at up to 50% seed moisture did not reduce yield, and they were able to get that crop out 2 weeks faster than letting it dry down by itself. If they made the application at 60% seed moisture or higher, then they would begin to see yield reductions. We've done some things here looking at application timing, and we based our timing specifically on growth stages. So we would check the field daily to make this application to these plots just as soon as the field moved into this particular growth stage, which what we targeted was R6, R6.5, and then we had a later timing of waiting until all the pods, 65% of the pods turned brown. Just to show you some pictures of what the field looks like at the time of application. So this was R6, so still pretty green out there. R6.5, we see some color change beginning in the leaves. The pods have begun to have a color change as well. I'll show you some more pictures here in a minute of that. And then obviously here's when waiting 65% brown pod. In my experience, when we waited till it looked like that, we did not gain but about 2 to 4 days from treated versus untreated in time. So the earlier it was sprayed, the more time we gained on the back end. But the bottom line is this: will it hurt my yield? Yes, you can spray these too early. You will see a yield decrease. In our particular case, that left bar on the blue bar here on this graph was the R6 timing, R6.5 in the middle, and the 65% brown pods to your right. So in my opinion, I guess at the end of the day, it really depends on how early they're sprayed. The earlier that you spray them before they are mature and ready, the more yield loss that you would see. From a timing standpoint and getting that benefit, we have seen up to a 2-week benefit of earlier harvest, but across our data, that range is from 4 to 11 days. And then again, we did see some indication of yield loss by not allowing that seed to fully develop and reach a true R6.5. So let's talk about that description real quick. And this is just a reminder of where on the plant to look when we're making this growth stage determination. Indeterminate soybean varieties, which are the majority of the ones that we grow here in Mississippi, they will begin reproduction at the bottom of the plant and progress upward as the plant produces more nodes. So what this means, the most juvenile pods will be in the uppermost nodes of the plant. If you look at determinate varieties, which are a lot of the group 5s that we plant in various places around the state, these determinate varieties, they'll flower uniformly up and down the plant. So when you're making the growth stage determination, you can look anywhere on those particular varieties. So pods are the key. Leaf color, stem color, all that can be influenced by a lot of outside factors. And I've driven through fields where the stems and everything were as green as could be. The pods were brown and way ready to harvest, and then vice versa. So I always focus on the pods, and I always focus on the pods in the upper 4 nodes, just in the case of I don't know what variety that we're dealing with to determine whether it's indeterminate or determinate. So R6, let's talk about that real quick. R6 is when the seed have fully developed within that pod. if you can look at that picture on the left-hand side of the screen, the center seed is square. It is so large inside that pod that it's pushed up against its neighboring seed, and the edges have squared off. So that seed has completely filled that pod cavity, won't get any bigger, and at this point, it will start receding in size and going the other way. So when we're looking at harvest date application, Drew mentioned irrigation termination, R6.5. There's a lot of things that we terminate at R6.5. So that top pod in the right-hand picture is an R6.5 pod. If you were walking out in the field, you would still see green pods, but they would begin to take on this yellow tinge of color. The key factor, I guess, is to shell open this pod and look at what the seed looked like on the inside. If you shell open an R6.5 pod, that seed will turn loose of the pod wall. So that protective membrane that is between the pod wall and the seed will separate from the seed, and the seed will be really shiny in color, but you notice it still has that greenish yellow tinge to it compared to those R7 and R8 pods. Lastly, I'm going to mention a couple things about grain quality. I really enjoyed Dr. Wilkerson's presentation earlier at the work that she and her co-PIs are doing. Here's some of the stuff that we've done, and really truly this one here was just by happenstance. We had a planting date trial in the field back in 2018. She mentioned all the rainfall that we got in 2018. So every chance we got that year, we pulled samples and sent them off to Mid-South Grain to have the overall damage scores evaluated, just to see what we had in terms of the different trials that we had in place. So again, this was a planting date test. You see that we had early, which would have been April, and late, which would have been late May. Planting dates, we see the yield value. We had a clear separation in yield. That's exactly what we expected there. Greater yield for the earlier planted beans. Interesting, we had 21% damage for the early planting date, and then not quite 5% damage for the late planting date. So a big difference in that. And what I attribute that to is the specific growth stage that those crops were in based on the weather events that happened. So the later planting date had more juvenile pods out there. The earlier planting date had more mature pods out there and suffered more damage just simply because of the growth stage that it was in. But interesting to put the dollar value to it. So we looked at the deductions of it. Our Ag Econ group helped us with this, and then we looked at our net return above treatment. What that means is that even though we had 21% damage, we did have a greater yield in our earlier planting date. So we took more of a deduction, but we made more money at the end of the day because of that greater yield value. Another thing that I'll mention, I guess in the fungicide yield data that we were talking about earlier, as a part of a quality project that graduate students are working on. So that particular project, we're looking at automatic fungicide applications at R4. And then we have separate trials in place where we're looking at harvest date applications. And then what we're doing is we have harvest delays associated with those various management practices. So we're not necessarily focusing on the management practice, but we're looking at the yield and quality based on harvest delays as a function of each one of those management strategies. So the column that you see here listed as normal harvest, that would be whenever everything is ready to go, we're harvesting it. And then we're letting other plots with the same treatment sit in the field for 2 weeks, 4 weeks, or 6 weeks delayed. And we have another layer on top of that too. We have a planting date component to that, which is a late April and a late May planting date. So essentially, we have a lot of treatments that are out there, and then we're doing a lot of harvest delays where we'll be harvesting plots from September through Thanksgiving. And our design behind that was just an effort of hoping that we catch adverse weather conditions that will help us, determine, do these management practices impact what we're seeing on poor quality or yield losses? We have not received all the data in yet from our grain quality. We're doing grain quality and also germination tests. We're still waiting on some of that data to come in for 2020, and then we'll analyze it. Here's our yield, though, across 2019 and 2020, and I was very pleased to see that stair-step pattern in that. So we do have a clear yield loss each 2-week delay that goes on. So obviously we have something going on whether it be, quality issues or shattering or things like that, which we have all those data collected as well. So stay tuned for the final of this project. We'll be releasing this just as soon as we get the data and get it analyzed. Dr. Wilkerson also mentioned, the work that they've done, looking at a lot of varieties and their responses for grain quality. We also began to do that in all of our on-farm variety demo plots each year. We're waiting on those to be returned, and we'll release the publication for 2020 just as soon as we get it. But each year we have these sets, and we'll have the quality at each location and the overall damage scores as well for producers to be able to use these in making variety selection decisions. Dennis mentioned the variety selection tool. That's a great benefit for helping. I know there's a lot of variety decisions that are being made, have been made, or are being made right now. Just here's some links to some different documents and websites that would help with those. Again, a special thank you to the board. They fund my program and everything that I do, and I couldn't do what I do without their support. And of course, thank you to all the seed companies who participate in a lot of our variety demonstration efforts as well. So that will wrap up what I have, and I have this slide here. So this is the code that Dennis mentioned at the beginning. This is what you will need to try to scan to receive your credit today for the CCA. And again, like he mentioned, if it does not work for you, there's his email at the bottom. If you need his phone number, I'm sure a lot of us already have that as well. So with that, I'll turn it back over to Carly or Dennis or whoever needs to wrap this thing up.
Sounds great. Thank you so much, Trent, and for all the other presenters with your time. There is certainly a lot of really exciting research taking place at Mississippi State, and we are thrilled to see the 2020 findings and what's included and what will be happening in the future. Due to time, if the presenters wouldn't mind to please add their email within the chat box. And for those that may have questions, please feel free to email them at that time with anything. So if you could, again, presenters, please just drop in your emails or the best way to contact you in the chat function. And for any folks that would have any questions on finding the chat or anything like that, please don't hesitate to reach out. And as those are coming through, again, as Dennis had mentioned, this presentation has been recorded. We will be distributing this recording here by email. We anticipate it being sent Monday, December 14th. So please keep an eye out for that. Feel free to share it with colleagues and everyone else. So it looks like Dr. Wilkerson, or Dr. Irby. So we've got everyone in. Perfect. All those email addresses. I'll give it just a couple more minutes in case you'd like to write those down or save them before we close out this meeting. But again, thank you all so much for your time and for working through us in this chaos. And I hope that each and every one of you have a wonderful holiday season. Thank you.